Have you ever wanted to control an army of insects against the invasion of an exotic plant or insect pest? Release a horde of minions that will tirelessly pursue specific weeds across your managed land. Today we'll discuss biological control, the science behind the practice, and both pros and cons of biocontrol in pest management. My name is Christy Gladham. I have worked in classical biological control for over 10 years. Welcome to the Colorado Department of Agriculture's Biological Control Series in collaboration with the USDA Western Sustainable Agricultural Research and Education Program. What is biological control? In short, it is a methodology for suppressing pest populations by using their co-evolved natural enemies. We'll need some definitions before diving in. When I say target pest, or host, that refers to the pest species we want to control. Pests that can be targeted by biological control are typically either insects or plant species. I'll mention the native range, which is where an organism is originally from or where it originally evolved within an ecosystem. This contrasts with the invaded range, which is the area where a pest has been either purposely or accidentally introduced. A biological control agent or biocontrol agent or agent is an organism that is a natural enemy of a pest species. These agents can be mites, insects, or pathogens, and they themselves can be either native or introduced in an invaded range. There are three types of biological control: inundative, conservation, and classical biological control. Inundative biocontrol, also called augmentive biocontrol, is when large numbers of natural enemies are released as needed to respond to periodic pest outbreaks. A good example is when ladybird beetles are used in greenhouses and gardens in response to aphid outbreaks. Conservation biocontrol is the practice of leaving refuge or native undisturbed vegetation for natural enemies to flourish. These are areas where natural enemies can live, reproduce, develop, or overwinter, and are protected from things like pesticides, tillage, mowing, or grazing. Their established populations are then able to quickly build up in response to increasing populations of pests. This is especially useful in monoculture cropping systems. It's a way to help keep natural balance in the landscape. Both inundative and conservation biocontrol work within the existing ecosystem and may target native species that can become pests through overabundance like aphids. Both can work well in agricultural and cropping systems. We'll focus on classical biological control in this video series, but there will be resources listed in the video description for other types of biological control and how you might incorporate them into an integrated pest management system. When we think of pest plants or insects, we might think about categories of organisms that aren't very pleasing in a broad sense, like thistles that have irritating spines or caterpillars that eat crops. However, classical biological control focuses on individual specific species. So instead of targeting thistles in a broad sense, a biological control program would target a singular species of thistle. Target pests of biocontrol fulfill four main characteristics. These are species that are not native in the area we are trying to control them, they are highly invasive, they are not easily controlled by other means, and they cause real economic or ecological damage. Why does it matter if the pest species we are trying to target is non-native? Most pests in the US were either purposefully or accidentally introduced far away from where they evolved in a whole ecosystem. These pests may have been removed from all their natural enemies and are in a new environment where there are not organisms that are specialized to feed on or exploit the pests for their life cycle. There may be no other species that directly compete with the pests. Plants that are being fed on by introduced pests may not have chemicals that will deter feeding or limit the pest growth. There will not be negative pressure on the pest population, and that gives the pest a competitive advantage against native species in the invaded landscape. Species in their native range have evolved over time with a full cohort, sometimes hundreds of organisms, that exert pressure and reduce the upper limit of a species population. This is good because otherwise a single species can proliferate out of control and destroy the ecosystem. It's all about balance within nature. That sometimes ebbs and flows but has an equilibrium over time. For a non-native pest introduced into a new environment without any of those checks and balances, the population can quickly displace natives, compete for resources, and destroy the balance of the ecosystem with other cascading effects. Classical biological control purposely and strategically introduces non-native organisms into an invaded range to reunite with their co-evolved host, who has become an invasive pest. Types of biological control agents might include pathogens, predators, and herbivores, or parasitoids. These could include bacteria, fungi, mites, nematodes, or insects. Classical biological control is an attempt to restore ecological balance in a landscape and place pressure on pest populations. Since this is a natural balancing act, biological control is expected to only suppress pest populations, never fully eradicate the pest. The process of finding and identifying good candidate species for classical biological control begins with a field study of the pest species in the native range. Scientists will observe and catalog the whole range of organisms that utilize the pest for their life cycle through feeding, parasitism, or development. This can include hundreds of candidate organisms, which are carefully narrowed down by their host specificity and the damage they cause to their host. To be considered as a biocontrol agent, an organism must be host-specific. That is, the biocontrol agent must feed and develop or complete its life cycle only on the target non-native pest species. If an organism is known to be a generalist that can feed on and develop within a large and varied host range, that organism would not be considered for classical biological control. The field researchers identify all of these organisms that feed on, parasitize, or live within the target pest species. The scientists also form a list of potential hosts, including the target pest species and other highly related species in the native and invaded range. These will help test the host specificity of the potential biocontrol agent. The list of test hosts can also include non-related species that are economically or ecologically valuable, which we would not want natural enemies to target. These two lists, the potential biocontrol agents and the species to test the agent's host specificity, are submitted to a technical advisory group, or TAG. The TAG, made up of subject matter experts, will then evaluate the list to make sure the biocontrol agents are thought to be host-specific and the most robust list of potential target hosts can be tested. Once the TAG has approved the list, very stringent laboratory and field testing is carried out to test the assumption of host specificity. Testing of biocontrol host preferences begins with a very conservative test called a no-choice test. This evaluates the response of potential biocontrol agents when given only non-target hosts. The best case scenario is that the agent ignores the non-target host being tested, and that host can be excluded from future tests. If an agent feeds, lays eggs, or develops on a non-target host, then that host is used for testing preferences when the agent is given the choice between the non-target and target host. Usually there's a very clear preference for the target host, but if not, then even further testing can be conducted where the agent is given multiple host choices in the laboratory. These host preference tests can then be conducted in cages in the field. If there are any questionable results, there may be further testing within the native range to resolve true preferences when the agent is in an open field. All of these tests will help tell us if there is potential for non-target effects and exactly how specific a potential biocontrol agent is to the pest species we want to target. A relatively new method is the testing of chemical ecology of hosts to test the biocontrol agent's attraction or repulsion from certain host chemical cues. This is really useful for testing the risk to threatened and endangered species without having to utilize those TNE species directly in lab testing. Either during host testing or after, permits to move the biocontrol agents into the USA can be issued by the USDA APHIS PPQ program. At first, the biocontrol agent will only be allowed to be kept in special quarantine facilities with strict protocols to prevent any release of the agents into the environment. Further testing will occur in quarantine to ensure the agent is the right species and not harboring pathogens. Scientists also look at life history and seasonal timing so we know if the biocontrol agents will be able to establish in the invaded range. When testing is complete, the research scientists will submit a petition to the TAG to be able to field release the biocontrol agent into the invaded landscape. The TAG will review all of the science and either request further experiments or give a recommendation to either release or not release the agent. Researchers must also work with the U.S. Fish and Wildlife Service for consideration of the potential impact on threatened or endangered species. There is also tribal consultation with sovereign indigenous nations to assess impacts on species of cultural importance. USDA APHIS PPQ will evaluate the outcomes from laboratory and field experiments, the TAG findings, U.S. Fish and Wildlife recommendations, and tribal consultations to reach a final decision. At this stage, biocontrol agents are either issued a permit for release, an environmental impact statement is requested, or the project is discontinued. Once approved, the USDA APHIS PPQ permit for field release is reviewed at the state level. Individual states have jurisdiction over what can or can't be released inside state boundaries. The overall process for identifying, testing, evaluating, and approving a new biocontrol agent can take a minimum of several years and up to a couple of decades. The intense process, standards, and collaboration used to assess host specificity makes modern classical biological control remarkably safe. Over 500 classical biocontrol agents have been released worldwide. In over 99% of cases, host specificity testing was successful in predicting non-target effects. Only 3 biological control agents worldwide have shown non-target effects that threaten native plant populations. Those non-target effects were predicted before release, but in those cases, the non-targets predicted to be affected were not valued in the same way we value our natural resources today. What can be expected from biological control? Once released into the invaded range, biocontrol agents can take time to establish significant populations. Sometimes, due to unforeseen circumstances, the population may never establish to the point where the biocontrol agent can cause significant suppression of the target pest. Some of the issues with establishment could include phenological or seasonal mistiming between the biocontrol agent and the target pest within the invaded range, predation or parasitism by native insects that the introduced biocontrol agent has no defenses against, novel bacteria or fungi that impact the biocontrol agent, or even divergent rapid evolution of the target pest in the invaded range that make it less suitable as a host for the biocontrol agent. If the biocontrol agent establishes well, that means it overwinters and starts reproducing right away, it may take 3 to 5 years to see results or reduction of the pest species. Sometimes full impact may not be realized for a decade or more, depending on the pest pressure. Results are often only noticed if careful monitoring or observation takes place, since we are talking about landscape-level suppression of the pest species over many years. One benefit of biological control is that it is self-replicating, so once established, biocontrols will persist in the landscape, providing continuous suppression of the pest. Biological controls can also co-evolve with their target hosts. In the case of misapplied herbicide, plants can develop resistance, and the formulation of that herbicide will no longer work. With biological control, as the target host develops toxins or defenses to the biocontrol agent, the biocontrol agent can develop antidotes or other strategies that compensate for the increased defenses of its host. This is also beneficial, as this evolutionary arms race is part of what makes biological controls so specialized to their hosts. In some cases, those defensive chemicals from the target host are incorporated and necessary for the life cycle of the biocontrol agent. Tying into that, another benefit of classical biological controls are that they are incredibly host-specific, meaning they can be released in areas that are more ecologically sensitive and they will not impact other species. This can be greatly beneficial in sensitive areas where pesticides or disruptive mechanical means of control are not possible. Biological control can also self-disperse into areas that are inaccessible for other means of control, like steep cliffs or very remote areas without roads for equipment or people. Biological controls are extremely cost-effective as a control strategy. A review in 2020 showed that for every dollar put into a biological control program, we can see between $800 and $7,400 of return on investment. This is based on the initial cost for research, release, and monitoring for establishment, but once established in the landscape, biological control provides landscape-wide suppression of pest species with no additional or yearly costs, so that return on investment grows year after year. Biological controls can reduce the need for pesticides by lowering the population of a pest species. This provides not only an economic benefit by reducing input or production costs in agriculture and land management, but also reduces pesticide exposure risk for workers and reduces pesticide usage in the environment. All management tools come with some risk, but there is also a huge risk if we do nothing. Biocontrol offers a low risk and high-reward management option to land managers. A really robust toolbox of control options is needed if we have any hope of managing invasive species. Biocontrol offers a safe, cost-effective method to add to our control toolbox for long-term management of widespread invasive pests. I hope this video has introduced you to the science, methods, and possibilities of biological control for invasive species management. There will be a list of resources and sources in the video description for you to learn more about biological control. Thank you for joining me today.